of native areas.
Mm.
And a lot of it is from overexploitation. Things are harvested or over harvested, resulting in land that results in the invasive species coming in. Inbreeding depression is something that I think is something that's becoming more and more problematic, especially when I'm out there looking at native plant populations and looking at seed production. You're basically dealt with a landscape that is not continuous anymore. It's very fragmented. So you have these little tiny islands of native plants, and there's essentially no gene flow from those. And so you're getting some inbreeding occurring, lack of seed production, which is basically resulting in those losses. Range shifts, there's simple changes in habitat use, in the climates. If the temperatures just aren't quite right, plants will start to move, or they won't survive and persist in the areas that they were historically found in. And I mentioned habitat loss and, of course, land use changes. And I wanted to provide one example that kind of ties into land use changes. This is a seed collection that was supposed to be Betula pumila. So many, well, I think it was back in 2014, I was trying to make some collections of Betula pumila in Iowa. It's listed as a T&E species in Iowa. And so there was, this work goes into looking at herbarium specimens that to figure out where it's been documented. And if you look at this example here, there was a number of reports of herbarium specimens that pointed to this specific site. And one of them was collected by Mark Leschke, who basically said this was one of the best fens on the Iowa surface. Numerous rare species are noted, and approximately 500 plants of Betula pumila in the northern part of the fen. And then later on, it was recorded again in 1987 by Diana Horton, and then again by Mark Leschke, and then Jeff Nicola in 1990. And all of these folks had documented that there was, thousands of plants in this fen. So it was in 2014 that I visited this site, and when I first arrived there, I thought something's not right. I must be in the wrong location. So I double checked my coordinates, double checked the road maps, and sure enough, I was in the right location. And what happened was is the adjacent land to this fen, shortly after 1990, had a area excavated to channelize a runoff from the adjacent field. Well, what happened was is that channeling basically completely changed the hydrology of this site. And within short time, by actually it was 2015, sorry. In 2015, when I visited this site, I could only find 3 specimens of Betula pumila, and this site was completely overrun by giant ragweed, reed canary grass. So it had completely been destroyed. So it doesn't take much for things to happen over a very short period of time. So what can we do to try to save these valuable resources? And there's 2 ways. One is it's called ex situ conservation, and the other one is in situ conservation. And basically, what that is we are either taking materials out, which is the ex situ, or we're protecting the materials that are there through particular means, typically like designating them as a state preserve or park or something like that, where we will not develop it at any point in time in the future. Now, there are some good pros and cons on both of these, and so this is the green page. That's the pros or the advantages of doing these types of conservation methods. With ex situ conservation, once we've taken those seeds or plants out of that environment, they can be easily monitored. So we can keep an eye on them out in my field plots, just out the door here a few feet, and make sure that they're being well taken care of. The conditions are typically controlled, so we can, if it's a drought year, we can water. If there's pest pressure, we can alleviate those pests with particular management strategies. The other thing is once we have these resources in an ex situ gene bank, we can distribute those. So there's always people out there wanting to do research on plants. And so sometimes it's hard to get those. But I always tell people we're kind of like the Amazon for plants, to be able to distribute plant genetic resources to those folks. Um, the advantages of in situ is that it does allow for natural evolution. So it's over time, things will change and shift a little bit, and the plants will adapt to what they're being dealt with, as opposed to the ex situ, where you're taking basically a snapshot of a particular site and collecting the genetics at that particular time. The other advantage of in situ is the fact that you can protect multiple plants or numerous species and those communities that are there. You can't always do that with ex situ conservation. Now, the disadvantage is, when we're doing ex situ conservation, sometimes there's less genetic diversity. Very, very rarely is a person going out and sampling every single genotype that's out there in a particular population. Sometimes when we bring these plants back and try to cultivate them, even though we're giving them everything that we think they need, sometimes the soil types aren't quite right, the hydrology is not quite right, and sometimes it's a pH thing. So there's often cultivation challenges involved. The other thing with ex situ samples is that when we're storing samples as seeds in our seed bank, we don't always know what the longevity is. How long will this species or this seed sample last in storage? They are living individuals, they are respiring, so at some point in time, they will perish, unfortunately. So that's a tough thing to try to deal with, especially when you have so many different plant species in the world, and trying to figure out how best to curate those. Um, in situ has some disadvantages in the fact that you have to manage those properties. I am not a huge believer in leaving it alone and walking away. There's too many things that our natural areas are being faced with, and so there does need to be some close eyes on those, and requires some management from time to time. The other thing is that even though a site may be protected, where it's not going to be developed, there's still the chance for adjacent properties to be developed. And that's when people will start planting other plants, often from cultivated unknown origins. So then you're basically exposing the preserved area to genetics of different plants and from different origins. The other thing is that extreme weather events can happen at any point in time. So, flooding, I've seen wiped out some natural areas. And of course, there's other examples as well. So that brings me to, talking about exactly like what I do here, and that is the ex situ conservation of plants. And the organization that I work for is called the National Plant Germplasm System. And this is a coordinated effort by the USDA, and it's basically a network of gene banks across the US. There's 20 in total gene banks, and if you went to each one of these sites, we're all doing kind of the same thing of maintaining these plant genetic resources, but the crops are very different. So if you went to Ames, Iowa, you would see a lot of corn, carrots, melons, sunflowers. And then I work with the herbaceous plants, a lot of the forbs, and then of course woody plants. If you went to Davis, California, you'd get to see the walnut collection or the grape collection. If you went to College Station, Texas, you'd get to see the hickory collection. If you went to Geneva, New York, you'd get to see the asparagus, the tomatoes, the apples. Collections. And so each of these sites maintains a specific set of taxa. And if you look talked about gene banking in general. Across the world, there's about 1,700 seed banks that exist globally that are basically maintaining about 6 million accessions. But what I like to mention here is the fact that the MPGS is tasked with maintaining these for in perpetuity. And so we're always monitoring the quality and the quantity of a seed sample. And if it falls below a critical level, that's when we start to execute our controlled pollinations so that we basically fill that seed jar back up with what the genetics were, or at least very close to what it was originally. So our whole purpose in the National Plant Germplasm is to conserve these genetically diverse uh, genetic resources and to curate the information. To me, that's the really fun part is knowing, where was it collected and who collected it and how long ago, what was the habitat like. So a lot of that information is pretty neat to try to extract and figure out and making sure that we're maintaining that. So if somebody asks us, 50 or 100 years from now, we can provide that kind of information. Once we have all these materials, we like to encourage its use to making sure that it's available for crop improvement and product developments, and of course for research. So if you were to come visit me in Ames, which anyone is welcome at any point in time, you would see a number of fields, and usually you'll see our pollination tents out in the field. And so if you looked at the screen, you can see these little tiny squares spaced out evenly from this aerial view. Those are our pollination tents that we will basically restrict pollen flow so that nothing is coming in and nothing is going out. And of course, we have a very large seed storage facility. And then so you would see lots of jars or packets with seeds. And then, of course, we have some fields that are maintained as plants. And the example on the bottom is a picture of our willow collection. Historically speaking, our willows were maintained as clonal plants in the field because we didn't really think that we could preserve seeds of willows because they were deemed recalcitrant. But it wasn't until a few years later we realized that seeds were actually orthodox. It's just the fact that they have to be handled in an extremely short period of time. We're talking hours. So these seeds are basically harvested, processed, and in the freezer within a matter of 24 hours. And then, of course, we have liquid nitrogen. So the old saying that the colder is always better is not always true for everything, but for the most part, a lot of species that can be handled being dunked in liquid nitrogen will last for very long periods of time. So what do I do as a curator? So my whole job here is focused on managing a collection of accessions. And a lot of that includes assessing each species for their gaps. The map off to the right there is an example of the Kentucky coffee tree collection. This was work that I was involved with a colleague, Andy Schmitz, who's the director of the Brenton Arboretum in Dallas Center, Iowa. And for about 15 years, we were out and about collecting coffee trees across its native range. And so the cross hatching is the Elbert Little maps from the 1970s across the ecoregions. And then each one of those points is where a coffee tree collection was collected. So once we've acquired those samples, we're trying to make sure that all of the latitudinal and longitudinal gradients are basically filled in. We were trying to shoot for at least one collection every 75 miles or so. Once we get those collections, we database all those, making sure that we keep track of the number of specimens that were collected, the associated species, and so forth. And then once they're getting ready to be stored, we do an initial baseline viability test. And then about every 10 to 15 years after that, we're doing a maintenance test to see where the viability is. Then, of course, if we have sufficient quantities, I'm distributing those to researchers. So a lot of portion of my job involves reading the requests, determining whether or not to distribute the samples. If you took a look at the collection that I have, a large part of it now is Fraxinus or ash, and I assume all of you can imagine why. So back in 2009, when the emerald ash borer was introduced, that started a reactive process to start collecting germplasm across the U.S. So if you would have looked at the collection or holdings of Fraxinus 25 years ago, it was probably only about 24 accessions, and now we're well over close to 600 now. The other collections that we have, we're very well known for the Echinacea collection that we have, the Betula collection. Monarda is one that I've kind of taken a lot of interest in the last couple of years. And of course, and Salix has been drastically increasing over the last couple of years as well. The major management activities, I'm going to go through each one of these. These are the main things that we're doing on a daily or yearly basis with our accessions. So the acquisition phase to me is probably the most exciting part and enjoyable part. It's kind of this hunt to find a particular species in a particular area. And a lot of it has to deal with going back to very, very old herbarium specimens. I can't imagine doing the work that we do here without herbarium specimens. And I think it's really gotten a lot easier in the last 10 years because a lot of herbariums are now digitized. And now you're looking at Facebook posts. We're talking about iNaturalist posts. So being able to find unique locations for plants has really gotten a lot easier over the last couple of years. Once we've acquired these, the main thing is we want to try to image these the best we can. We'll take herbarium specimens ourselves and then submit those to regional and then the National Arboretum Herbarium. And when we're out there trying to acquire these, the whole goal is to try to get a representative sample of that population. And I always tell people, that we want the good, we want the bad, and we want the ugly. So just because something looks bad now doesn't mean it won't be valuable 50 or 100 years from now. And I think for a lot of people that's an interesting challenge. But the first slide that I showed is a very good reason why we want to do that. Because once we have those genetics in our holdings, that in return means that the likelihood of success when we do a restoration, if somebody's requesting these materials, that there will be some individuals able to establish at the time that they're being put out in the landscape. The documentation part, like I said, is very important. It provides a historical reference. It's basically very, very similar to what a herbarium specimen does. Except in our case, we're actually maintaining a living collection. When we're documenting these things, we learn from it every single time we're out there collecting. And I always tell people, I like, historically speaking, we used to go out and collect lots of species from a given area. And now it's kind of shifted more towards focusing on a particular species and then targeting multiple areas across a very large geographic area. And I've found that that's been more useful because we really learn more about that specific species. Exactly where does it like to grow? Where does it want to grow? What does it grow with? That plant community. And it always amazes me, like, when I think about when we're doing restoration work, not myself, but others, folks, how well are we really pinpointing where those seeds need to go or those plants need to go? Because you look at a wetland area as an example, you can be on a low shelf and you'll see a certain group of species, and then you go up 2 feet higher in elevation and the plant community completely changes. So it's really hard to know where we need to be putting these plants out in the landscape. So this is an example off to the right is an example out of our webpage. This was actually a collection in Douglas County, Wisconsin, I made a couple years ago. talking about what the species were, and of course comments. Sometimes the plants are pretty rare. We try to indicate how many we saw, um, and anything that might be useful down the road. Once we have these collections and they're getting stored, we have a couple options. We have a short-term storage where we have anything that's held at these refrigerator type conditions. Those are our distribution samples where we'll go into the cooler and pull and distribute. And then we also have a long-term storage, which is a freezer. Which, thankfully it's nice to have that freezer, and I wish we had bigger freezer space, because if we have to store things in the fridge, we recognize that they won't last as long. So if we were able to store them in a freezer, the longevity could be extended. Evaluations. It's always fun once we get these plants to see how do they compare to each other. So this is an example of some river birch. This was an evaluation study done trying to identify river birch that would tolerate higher pH soils. And when we collected these samples, we were also taking soil samples to see what the pH was, in hopes that we could identify some genotypes that would do better in the urban landscape and not be chlorotic, like so many are in the Midwest. So sometimes those characters may be phenological. Maybe we're looking for something that blooms a little earlier or a little bit later, or blooms for a longer period of time. And then, of course, agronomic traits, especially if you're a nurseryman trying to grow plants, you do need stuff to perform. The distributions, we typically distribute about 45,000 samples a year. If you look at the entire NPGS, our entire system distributes about 250,000 items annually. A lot of times that's those seeds, but we also are shipping out cuttings. Sometimes I ship out whole plants. Sometimes people don't want plants, don't want seeds. They just want the leaf tissue for genetic analyses. So there's always some interesting requests from time to time. And the great thing about that is that I'm always making connections. These people that are requesting the materials will have some interesting research projects and funding that they've gotten. A lot of times they'll report back to me a couple years later with some interesting traits that they've discovered. And of course, that usually always results in publications. And once we get publications that document the Ames number or PI number that was distributed, that gets tagged in our database. And so the other thing is that people will sometimes tell me like, hey, do you have this? Or, like, why, I see you don't have anything from this area. So that's the tool that I use for, getting that opportunity to assess those gaps. Where do I have missing collections that people need? And so I set a picture down at the bottom of some Salix pedicularis. This was a collection from central Iowa. These went to our state forest nursery. So this was 4,000 cuttings that recently got direct stuck in the ground for production. This will, I think, be the first time that the state forest nursery will be distributing a known sourced and species identified Salix collection. So that's always a good feeling to know that some good native plants are getting put back out in the landscape. Characterizations. So sometimes we're taking specific traits from these accessions. And if you were to take a look at the Monarda as an example, a couple years ago, we did a grow out of about 20 different populations. And I think we collected close to 35 different traits, looking at measuring those and then documenting those in our database. And once those are in the database, requesters can search those. They can look for a specific trait or even multiple traits. And basically, you'll enter in that data, and then you'll get a regurgitated list of accessions that meet that criteria, which for a plant breeder is those pre-breeding efforts that they already need to know for what they're trying to target in their program. So what's been done in Wisconsin? So this was kind of fun. I got to do a little searching through the database and counted out a total of 492 accessions from Wisconsin. The very first one originated in 1907, and then the most recent one was an Amelanchier. And if we looked at the most common genera collected, no surprise here, it was ash. But the next one was a little surprising to me, was cannabis. So the Geneva, New York site has vamped up collections of cannabis, and there's even some researchers in Wisconsin that are actively collecting cannabis, wild feral types, and then of course sunflowers. Some of the more recent trips that I've been involved with, one was in 2022, one was in 2023. The trip for 2022 was actually focused specifically on black ash. And so there was a little loop that was taken throughout the central part of Wisconsin. And, I've been kind of watching these. I've been talking with some of the DNR folks. I really need that kind of help of like, hey, it's a good year. And once we know it's a good year of seed production, that's when we drop what we're doing and we head out to make those collections. Because I think for at least 3 or 4 years, there was absolutely little to no seed production in black ash. So it took a couple years before we finally got that executed. And, we did this central part of Wisconsin because that was a huge gap. There had been no other collections made in that area before. So 7 sites were sampled. The sample size was 90 specimens in total were collected. So we did approximately 10 to 12 mother trees per site. When we're out there collecting, we try to sample, like I said, the good, the bad, the ugly. But sometimes we'll harvest really young specimens, middle age, old. Sometimes we'll try to strategically target ones that have early fall color or late fall color so that we have a good guess that we're getting lots of genetic diversity. Yeah, then 2023, some sycamore was collected, which is a species of concern in Wisconsin, and then also Kentucky coffee tree to fill that Kentucky coffee tree gap. What are the future targets? This is a list of species that are kind of on my mind. And these are on my mind because they're of interest and they're gaps in our collection, or it's because someone else has prompted me to take a look at these. And so I'm going to go through a couple of these, or actually each of these. The first one is the American highbush cranberry. So if we look at the collection that's in the MPGS, there's a total of 27 collections. Only 2 of them are from Wisconsin. And so to me, that indicates there's a large area that could be sampled in the near future. And one of the reasons why this is kind of on my mind is because of its confusion with the European highbush cranberry. And the best way to identify or separate these 2 varieties is looking at the petiole glands, which I think is still can be a challenge. But the major distinction is the fruit flavor. If you ever get a hold of a European cranberry fruit, you will know it immediately. It has a very awful taste to it, and so you'll be very hesitant to ever try one again. But the American one does have some astringent tendencies to it, but it does have a hint of sweet. The other reason is that it's under attack by the viburnum leaf beetle and also a crown borer. So these natural populations are under attack. Some of you may be aware of David Tork at the University of Minnesota. He's a researcher and grad student that's working on the genetics of the European and American highbush cranberry. And so a couple years ago, a lot of samples were collected across the upper Midwest and submitted to the lab. And they did these, they figured out the genetic markers on these to figure out if they could separate them. And the good news is that they figured out that hybridization was really rare. Out of like 295 samples that got submitted, only 6 of those were identified as hybrids. I thought that was a little surprising. I wasn't quite sure, but there's definitely plenty of native populations that are within close proximity to European plantings. Unfortunately, a lot of nurseries have incorrectly distributed when they've marked or tried to sell plants as American hybrid cranberry, but they were actually European. So when I said they're often confused, they are confused a lot, and it can be challenging to separate these out. The great thing about the David Tork's work is that they have a genetic test that's basically in the process or near being completed, a genetic test that can definitively tell you whether it's American or European. And so that's a nice thing to have here in the future, and I hope to pass that around here in a short time. And also because of the European spread, it's taking up space in basically the same habitats that American high-bush cranberry would. So I think there's room to make a few more collections out of Wisconsin on this species. Pediomelium argophyllum is the silvery scurf pea. This is a species that has a very minimal history. The very first sample that was ever collected was back in 1954. Basically 70 some years later, a second sample was collected out in the Loess Hills of Iowa. And it's been brought to my attention not only from Kevin, but it's also from a number of folks in Iowa looking at this species. A lot of folks that I communicate with have indicated there's essentially little to no seed production. And that was evident in the sample that we got in 2023. Bill Zales was the one that made that collection for me. And I think he indicated he had sampled approximately 400 plants. And in that sampling of 400 plants, we only ended up with 126 seeds. So perhaps it doesn't produce seeds very often because it is rhizomatous. It will form these very dense colonies, as you can see in the picture here. But it is not represented in the nursery trade. None of the seed companies for native plants or prairie plants, you can't find this species. So is that because it's difficult to grow in cultivation? Is it just because it's not producing seeds? That's another reason why we're going to be looking at this in the near future. And it is a species of concern in Wisconsin. And so hopefully we can figure out what the best method is to get seed production and potentially see this species in the trade someday. Rock elm. I'd love to know how many people are aware of this species. I think it's not reported very well, largely because it's also difficult to identify. And when I say it's difficult to identify, I'm specifically talking about the genotypes that do not have the corky stems. That is not a reliable trait to identify this species. And I think it's a great species because of its wildlife value. The seeds that are usually shed in the first week of June are highly attractive to chipmunks, mice, turkeys, birds. A lot of times birds will steal them out of the trees before they even reach maturity. So it's pretty cool to see that. Oops. But if you're out in the springtime, usually I would say about the end of or middle of May is when, or early part of May. Maybe for you guys, it'd probably be second week of May. You can find these flowers that are pendulous, which is very, very distinctive for this species. If you look at Ulmus americana or rubra or even pumila, they have a very, I lost the word, clustered formation of flowers. And so that's very different from the Ulmus thomasiae that's pendulous. So that's one easy way to identify it. And I wanted to mention that, in Boone County, which is central Iowa here, there was a floristic study done in 1915, 1985, and also in 2010. And all three of those reports indicated that Ulmus thomasiae was rare. And one of the reports basically said only one specimen found. And so I thought, well, it'd be fun to go find that. And so I went out in the springtime, and sure enough, within a matter of about 3 hours, I was able to find about 75 specimens. And sure enough, only two out of those 75 specimens had the corky stems. And so it is difficult to identify. And honestly, I struggled to tell the difference between rock elm and American elm unless it has the fruit or the flowers. A lot of people have asked me about this. Do you think that rock elm has some Dutch elm disease resistance? And so it kind of sparked some interest. I think it was 2023, or maybe it was last year. Peter Moore from Iowa State University was teaching a class, forestry class, and we got the class to come out and do a lot of core sampling on both American and rock elm specimens. And sure enough, the specimen here that I'm had my picture taken with was 21 inches in DBH, was calculated at 300 years of age. So if something can live that long, I would say it's definitely resistant in my book. And so this was a snapshot of some forestry inventory data from Wisconsin. And as you can see, rock elm was basically the percent total was calculated at 0. But I'd love to see if people can have some eyes out in the field just to see where is it at, and is it just the fact that there's no corky specimens in Wisconsin at all? Is that reason? And we just hoping to get some people's eyes on there just to see what's out there. It's also another species that's not really represented in the nursery trade. Salix pellida. So this is another work that's been done from the University of Minnesota. So John Larson is a grad student there working under Dr. Brandon Miller. And their work is trying to look at basically, what's going on with Salix pellida. There's very few specimens, very few populations. Can we learn something about, why is it declining in nature, and then try to get some of these materials preserved, both ex situ and in situ, in collections. From a historical perspective, there's only been one collection ever made from Salix pellida, and that was last year. And there is none from Wisconsin. It is marked as an endangered species in Wisconsin, and there's, I believe, only 2 records, and it hasn't been seen in the last 20 years. Maybe it's still there and it just hasn't been noticed, or maybe it's gone. But it's from the extreme northern part of the state, up in the Apostle Islands. Lastly, Amelanchier. For a long time in my career, I've kind of shied away from Amelanchier just because they're taxonomically challenging. But once you dive into it, it's not that hard. And so in Iowa, in the last year, a lot of the non-Amelanchier arborea, so like Alnifolia, Humulus, Laevis, and interior have been assessed and looked at, and basically, they've been reclassified as either endangered or threatened, based on what's been found. And so there was a lot of collections that I did in the last couple of years on Amelanchier. There's in total 185 collections in the NPGS, but there's only one, which was the more recent one from Wisconsin, that Amelanchier bartramiana. Some of these are apomictic, so that means even if you collect seeds off of them, there's going to be genetically identical to the parent tree. But one of the things about these Amelanchier is the challenge of getting the fruits. Getting those seeds harvested is a huge challenge. The birds, as soon as birds start to see these starting to turn color, they're starting to get stripped. And so it's hard to beat the critters to these plants. And so I think it'd be fun to see what's out there in Wisconsin if people are interested. Um, so in potentially making some collections for the seed bank. So my last slide here is basically my cry for help is that preservation help is always needed, always welcomed. And that main part of that sometimes is just people's site recommendations. I mentioned earlier my work with looking at herbarium specimens is huge and it's key, but it doesn't always provide me an insight on what's currently there. Um, so sometimes word of mouth is the best sort of information I can get from somebody, especially if they have a good memory and they recall where it was and about how many specimens there were. But I also need insights on phenology. Um, Before this discussion even started, Kevin and I were talking about the pasque flowers, so if they were in bloom yet here in Iowa, and when might they be in bloom in Wisconsin? And sometimes for some species, it's fairly easy to kind of know where things are at, but for other species, um, they can be weeks different. My friend right now is down in northern Arkansas, and the elms are already have mature seeds. So, and here in Iowa, our elms haven't even broken bud yet. So there's some huge gaps in phenology, especially the further away from home a person gets. And then, as I mentioned, like with the black ash, is knowing what the fruit production levels are. So I can't be spending my time driving around to faraway locations to find that nothing has been produced or it was a very bad year. So I really rely on local help to tell me, what sort of fruit production levels are for that year. And then if folks are interested in even going a little bit further, I'll always welcome assistance in sampling. I think it's a great part to be a part of. Knowing that your efforts or our efforts together, whether it's together or singles, is knowing that that sample might be there for many, many, many years down the road. Think about the herbarium specimens that you go to and seeing that person's name on that specimen and that they had collected at that location. It's almost identical to our collections, except they're with actual living seeds or plants. So I hope I've uh, inspired some of you to take interest into ex situ and in situ conservation. And I've listed my email there if anybody wishes to reach out to me in the future. And thank you for your attention.
Thanks, Jeff. That was awesome. So if people have questions, why don't you type them in the chat, or if you can't access the chat, raise your hand and I'll call on you and you can mute yourself. I've got a bunch of questions. I'll ask one and then maybe give some people. There's one question in the chat already, but give some people some time to write. When you say assistance in sampling, how should people send stuff to you?
Well, I think that the first point is talking and having communications about what species that would be. So I mentioned there's 492 collections in Wisconsin. There is a small chance that someone might say, "Hey, I got this sample and I collected it here." There is a small chance that I could say, "Oh, well, we already have one." So
Mm.
first step is having the communications and visiting with me or a curator in the MPGS and saying, this is what I'm really interested in. This is a species that I'm passionate about. Maybe you have a connection to a specific species or a specific site. And that would be the first step. And then the second step is, can we logistically and realistically preserve that? Um, Kevin, you and I have already talked about the logistics of the silvery scurf pea.
Mm-hmm.
That's going to be a challenging one. And in that case, it's a challenge of being able to find a site that still has a sustaining population where there is seed production. And I think with this case of the silvery scurf pea, I think that you might be potentially looking at one plant at a site and it's just not getting pollen flow anymore. So therefore you don't have the seeds. So that's one scenario. The other scenario is can we logistically store the seeds using uh, a refrigerator, a freezer, or liquid nitrogen? There are some species, like oaks are an example, or magnolias are examples. Those are recalcitrant species where those seeds cannot tolerate being desiccated and dried down and frozen. And so those are species that we have to maintain as plants in the field. And so if we can, I'd prefer to try to work with species that can be stored. But sometimes we have to say, hey, this is an important species and we need to do something. So it just means that there's a lot more work involved. So
So
once we've gotten past those, then we can talk about like, hey, I can come up and help if you can get me, into the right place, or hey, if you're willing to do that on your own, here's kind of this things that you need to keep in mind that there'll be a checklist of when you're making these samples.
yeah, so don't just start sending you a bunch of stuff that you collect. Check in with you, make sure it's a
Yes.
good, you have the capacity to take it on, talk about a game plan to collect and when to collect and all that kind of stuff.
And today was actually a good example of knowing when to do the work. And so today was an example. I was cleaning a dahlia purpurea, so purple prairie clover. It was a seed collection that was first sampled in 2020. And we went down, targeted this site, made the seed collection, and we ended up with 46 seeds. Not what I would consider ideal, especially for a species that we know, can have a good abundance and that sort of thing. It was on a prairie remnant, and so we decided, oh, we'll hit it again next year. And about the same thing happened. We ended up with like 500 or 600 seeds. And then nothing happened in 2021, 22, 23. And then, or excuse me, in 22 and 23. But then last year, we made the sample, 27,000 seeds. And so it was just one of those things. It's just like, okay, we have something on our mind, right? But we just have to know, is it the right time to make that move? And the last thing I want to do is go to a site, you know, making some disturbance, making some collections, sampling from the plants, and then to come back and find out we should have just stayed out of there. So that's it, but that takes experience in knowing that. That's not always easy to know. Um, yeah. So
Yeah, no, that's good guidance. Okay, we have one question in the chat. David Egan asks, so, and maybe you answered this in the talk after he asked the question, but when you're talking about storing stuff, preserving stuff, are you talking about just seeds? You did mention cuttings as well, so it's everything, right?
yeah, I mean, like I said, cuttings, we can take cuttings and we can preserve those. And so with especially the ash and with the willows, those can be preserved in liquid nitrogen when collected in the dormant season. So we're usually out collecting the first week in January, making those samples. And so that is an option, is to store cuttings. Now, uh, the difference is the fact that you're sampling one plant, okay, and you're getting one genotype. As opposed to if I sample a seed head or infructescence off of a plant and I only sample one plant, I may get 10, I may get 50, depending on what species it is. I may get lots of genotypes because we know what the maternal parentage is, but the paternal, the father, there could be numerous fathers that donated to those genetics. So when I'm collecting seeds, I'm getting a lot of genetic diversity. So, but we can sample both. And in the case of willows, I've learned over the years that sometimes it's better to harvest, even though I could harvest seeds, it's better for me to harvest cuttings so that when I get those cuttings back to my facility, we can grow those out and control pollinate them. Because I've had too many instances where we would go out to a site in nature, collect the seeds, and we knowingly collect off of the right species, but a lot of the seeds that we get produce hybrid progeny, so they've crossed with something else. That's always a potential of concern. And I think for some species like willows, they hybridize a lot. So you need to know the species a little bit too before you start deciding how to sample it and what to sample.
Yeah.
But that's a very good question. Hopefully that was the right attack on answering that was what you're looking for, David.
Let's see. We have another question from Roberta. She says, does USDA expect collection emphasis to be on plants with likely commercial or conservation use, or are you quite free to make judgments on what you collect?
So it would be both. And I would say that, I do have what's called a advisory committee that's made up of university professors, nurserymen, that basically help advise what we should or should not be working on. And sometimes it's for commercialization, sometimes it's for conservation, and sometimes you can do one species and it hits both. So like the Monarda has been a very good example of a species that can be good both from a conservation standpoint and it's highly sought from nurserymen because of its pollinator value and use in the landscape. So it can be both. The other thing is like I mentioned with Kevin, is getting poked a little bit, getting asked about a particular species. If someone wants to approach me and say, "Hey, I really think this is under the radar. Nobody's looking at this." That's where I can step in and say, "Okay, let's figure something out. Let's do some work on it." So that's where that can go.
Okay, it's almost 7:30, but I have well, I have a few questions, but I'll narrow it to one. Something I've been wondering about, and maybe Jeff, maybe this isn't your area of expertise, so feel free to just say that. But what are your opinions on outbreeding depression? I know you guys said you have a lot of echinacea, for example, in your collection. Um, and when we're talking about planted prairie and planting things within its range and don't plant outside the range, and this fear of in like a prairie planting that something, if that planted prairie is right next to a remnant, then those two things are going to the echinacea, the pale purple coneflower, for example, from the prairie planting is going to cross with the remnant population, and that's going to make the progeny less fit because the ones in the prairie planting were raised in a nursery and aren't suited well for the remnant. So like I understand the idea of outbreeding depression sort of academically, but I wonder how often it actually happens in the wild. Do you have any opinions on that?
Um, that is an area of research that needs a lot of attention. And I think it's honestly can be a case by case basis. I was actually listening to a seminar just last week that was talking about looking at latitudinal shifts of three prairie species in Minnesota. And there was some differences when one species was moved from north to south for one species, but it was no change for the another species. So there's really is a lot of work that needs to get done there. And I do agree with you that, if we do have things moved too far out, that there's a less likelihood for them to adapt to the conditions of the area that they're being forced to be regenerating. Um, yeah, I mean that's a loaded question, and I would say, I'm always trying to think about the person that's trying to do these genetic studies 20 years from now. I think we're at a key point in time where we need to try to get a lot of these studies done now because, or maybe it's too late, depending on who you talk to. We've been moving plants around for a really long time, but there's still a lot of species that haven't been touched, and that silver screw pea is a good example.
Yeah.
Uh, but yeah, I mean, I noticed this, like with germination tests. I can tell you that if I take a particular species and it's from a very southern source versus central and a northern source, um, the viability scores will be the same, but I can tell you that the day at which they all pop is different across those gradients. And so it makes me think about like, okay, well, if we're taking southern genetics and putting them in a far northern landscape, are they going to germinate so far behind that they will never have the chance to establish? Or if we
Mhm.
take something that's far north and move it south, are we going to have a different impact where the seeds germinate right away and it gets overrun because that species is better adapted to those cooler conditions because it germinates earlier in the season? There's so many different discussions and arguments that can be made with this. It's really hard to generalize,
Mhm.
period.
Yeah. Yeah, I agree. It's hard to give broad guidance on
Um,
this stuff when it's really a species by species thing. Okay. Well, let's leave it there. Yeah, again, Jeff, this was really great. We really appreciate your time. And answering all these questions was great too. So, yeah, again, for people who are on, if you're in the Madison area, there's the social event in Fitchburg next week, and then in a little less than a month, we have Andrew Hipp talking to us about oak origins. So, thanks everyone for coming tonight, and we'll see you soon.